In the early 2000s, geneticists finally had the tools to ask a question that had been argued over for centuries, without any real data to settle it. Do the racial categories most of the world grew up using actually correspond to anything real in human DNA? A landmark study analyzed genetic markers from over 1,000 people across 52 populations worldwide, built specifically to test this question rigorously for the first time. What the data showed surprised even some of the researchers who ran it. It didn't confirm what most people on either side of the usual argument expected to find. This is what the actual genetic research shows about human racial categories, ancestry, and the deep, measurable history written into every human genome on Earth. For most of modern history, the assumption ran roughly like this. Humanity divides into a handful of large, biologically distinct racial groups, categories visible at a glance, rooted deep enough in biology to matter medically, socially, and scientifically. It's an assumption baked into old textbooks, immigration forms, and centuries of pseudo-scientific classification systems built by naturalists who never once tested the idea against actual genetic data because that data simply didn't exist yet. 18th and 19th century scientists ranked entire populations into rigid categories based on skull measurements, skin tone, and a handful of visible traits, then used those same categories to justify colonialism, forced labor, and centuries of unequal treatment, treating what were essentially guesses dressed up in scientific language as settled biological fact. Here's the first real crack in that assumption, and it came from a massive international research effort. In 2002, a team led by geneticist Noah Rosenberg, using data from the Human Genome Diversity Project, a project built specifically to sample and preserve genetic diversity from populations around the world, analyzed genetic markers across 1,056 individuals drawn from 52 populations, spanning every inhabited continent, publishing the results in the journal Science. Using a statistical clustering method called structure, which groups individuals purely by genetic similarity, with no information about geography or self-identified ethnicity fed into the analysis beforehand, the algorithm did find structure in the data. When told to sort people into 5 clusters, the groupings that emerged loosely tracked 5 broad geographic regions, Africa, Europe, and the Middle East, combined East Asia, Oceania, and the Americas. That result got seized on quickly by people looking for genetic proof that traditional racial categories were real. It also badly misrepresented what the study actually found. The clustering wasn't discrete. It was a statistical approximation layered on top of something far messier and more continuous underneath. Human genetic variation that changes gradually, population by population, with geographic distance, and shows no natural, sharp boundaries anywhere along the way. Force the software to sort people into 5 groups, and it will find 5 groups. Force it to sort into 10 or 20, and it finds different, equally defensible groupings instead, because the underlying reality isn't boxes, it's a gradient. Here's the deeper layer that made this finding so significant. Long before Rosenberg's study, geneticist Richard Lewontin had already calculated, back in 1972, that roughly 85% of all human genetic variation exists within any single traditionally defined racial group, not between different groups. 2 people from neighboring villages in the same country can be more genetically different from each other than either one is from someone on a different continent entirely. Subsequent, far larger genomic studies using modern sequencing technology have consistently confirmed Lewontin's basic findings. The genetic differences separating broad racial categories are real, measurable, and small, a thin statistical signal riding on top of an ocean of shared human variation. So why does any geographic genetic clustering exist at all if the categories themselves are this blurry? The answer traces back to how humans actually spread across the planet. Modern humans originated in Africa roughly 300,000 years ago, and for the vast majority of that time stayed there, accumulating deep genetic diversity across the continent as populations mixed, separated, and adapted to dramatically different regional environments over hundreds of millennia. When relatively small groups began migrating out of Africa, starting somewhere around 70,000 years ago, each migrating group carried with it only a fraction of the total genetic diversity present back home. Geneticists call this the founder effect, and it repeated itself again and again as those descendant groups continued pushing further outward, into the Middle East, then Central Asia, then splitting further into Europe, East Asia, and eventually crossing into the Americas. Each of these successive migrations acted like drawing a smaller and smaller sample from an already limited starting pool. A 2005 study led by geneticist Sohini Ramachandran, analyzing genetic diversity across 52 worldwide populations, confirmed this pattern with remarkable precision, finding that genetic diversity decreases in a smooth, measurable, almost mathematically predictable way the further a population's ancestral migration route traveled from East Africa. Populations in the Americas, having traveled the furthest through the most successive founder bottlenecks, showed the least genetic diversity of any region studied. This single pattern, sometimes called the serial founder effect, explains a huge share of why continental populations show any detectable genetic clustering at all. It has nothing to do with separate biological origins, and everything to do with the mathematics of small groups repeatedly splitting off from larger ones over tens of thousands of years. Picture a small band of early humans, somewhere in the Horn of Africa or the Arabian Peninsula roughly 70,000 years ago, preparing to move into unfamiliar territory. They carry with them only a slice of the genetic diversity that exists back among the much larger, much older populations they're leaving behind. Their descendants will split again and again, spreading across Asia into Europe, eventually crossing a frozen land bridge into a continent no human has ever seen before. At no point does anyone in this chain of migrations experience a single dramatic transformation into a new race. It's an unbroken, gradual continuum of ordinary people having ordinary children, generation after generation, drifting slowly across a map over an almost unimaginable stretch of time. Here's the twist that most people never learn, and it directly undercuts the idea that visible traits reliably track deep ancestry at all. Traits like skin color, often treated as the most obvious marker of racial category, turn out to be a poor guide to overall genetic relatedness because these specific traits were shaped by strong, repeated, independent natural selection, not by shared ancestry alone. Research led by biological anthropologists Nina Jablonski and George Chaplin has shown that dark skin evolved as protection against intense ultraviolet radiation near the equator, while lighter skin evolved independently through different genetic mutations in different populations that migrated toward regions with weaker sunlight, where lighter skin allowed better vitamin D production. Populations in southern India and Aboriginal Australia both evolved dark skin independently through separate genetic mechanisms specific to each population, despite having no close shared ancestry and having been geographically separated for tens of thousands of years. Skin color adapted to sunlight. It didn't track ancestry the way people assume. Disease resistance tells an even sharper version of the same story. The genetic trait for sickle cell, famously associated in popular imagination with a single racial group, actually appears independently in several genetically distinct populations across parts of Africa, the Mediterranean, the Middle East, and India, essentially anywhere with a long history of intense malaria exposure. Carrying one copy of the sickle cell gene provides real, measurable resistance to malaria, and natural selection favored that mutation wherever malaria was a serious, sustained threat, regardless of which broader population happened to be living there. The gene tracks a shared environmental pressure, not a shared racial category, appearing and disappearing across the map based on local disease history rather than any deeper unified ancestry. There's one more finding worth sitting with, because it reframes this entire topic on a genuinely human scale. In 2004, statisticians Douglas Rode, Steve Olson, and Joseph Chang, publishing a mathematical population model in the journal Nature, calculated something almost nobody expects to hear about their own family tree. Tracing back through the branching math of human reproduction, accounting for migration patterns and historical population sizes across every inhabited continent, they estimated that the most recent common ancestor of every single person alive today, on every continent, likely lived only a few thousand years ago. A figure far more recent than most people would ever guess. Going back further, to roughly 5,000 to 15,000 years, the model reaches what researchers call the identical ancestors point. A moment in the past where the ancestry of every person alive today, without exception, traces back to the exact same, complete set of ancestors. Not most of humanity, all of it. If a person living at that time has any descendants alive today, anywhere on Earth, they are a literal ancestor of every single living human being, regardless of continent, regardless of any racial category anyone might apply to their descendants today. Run the same math backward far enough, and every family tree on the planet, no matter how distant the branches look on the surface, eventually folds back into the same shared trunk. So when scientists actually compared the DNA of populations across every inhabited continent, the results didn't confirm the deep, separate racial categories many people expected walking in, and they didn't erase the real measurable genetic patterns left behind by tens of thousands of years of human migration either. What they found was something more precise than either extreme. Genuine, traceable genetic structure, shaped by founder effects, migration routes, and repeated independent adaptation to local environments, layered on top of an ocean of shared variation so large that the traditional boundaries between racial groups dissolve the moment you actually look for them in the genome itself. The map humanity drew of itself, sorting people into a handful of fixed, separate categories, was never really written in the DNA. The real map is a single, continuous, overlapping web of ancestry, connecting every person currently alive, back to the same small ancient population, and to each other far more recently and far more completely than almost anyone assumes. None of this erases the real, lived history built on top of those categories over the centuries, the social systems, the inequalities, the entire architecture of how societies have organized and mistreated people based on visible traits. What the genetic data actually challenges is the idea that any of that architecture was ever built on a solid biological foundation to begin with. The lines people drew were real in their consequences, and imaginary in their biology, two separate facts that turn out to sit uncomfortably and permanently side by side once you actually run the numbers.